Okay, this is Unit 7. This is going to be 7A, hopefully really quick. Basically what we have here is we have a spinning DNA molecule, which of course is not spinning. Let's see if there we go. Spinning DNA molecule, and the reason I put this one on here is I want to actually show the kids that these DNA strands don't really touch each other. They're hydrogen bond together. So we have the A's and the T's and the C's and the G's pairing complementary base pairs, but they actually share hydrogen bonds, 2 between the adenosine and thymine, 3 between cytosine and guanine. I also put it here because we're going to do a strawberry DNA lab. This is a picture of my kids a few years ago, and you can see DNA shows up very well. We have 46 strands in our normal cell after we go through DNA synthesis, each one about 6 feet long, but they're incredibly thin. But we can actually tease them out of strawberries and bananas, I guess, too. And then this one here is that they share half my DNA with a banana, which came from a 60-minute episode a few years ago. And it's kind of amazing to think about a banana and a human actually sharing DNA. But if you think about cellular respiration, if you think about mitosis, if you think about utilizing carbohydrates, lipids in terms of making DNA and RNA synthesis, that we have the same kind of code because we basically use our energy and create new cells the same way. So 6A, scientists, DNA structure and replication. History. For a long time we didn't know what part of the cell actually carried the genetic material. Lots of people thought it was DNA, other people thought it was maybe the protein coat, other organelles inside of it, but we actually ran some experiments to actually figure this one out. And the first people that actually were successful in determining this was Martha Chase and Alfred Hershey. What they did is they used a bacteriophage, which is a virus that actually affects bacteria, and the bacteria they picked was E. coli bacterium, a very popular bacteria because it's in fecal material and does make water not good to drink, as well as if we wash things in that water, not good to eat. So in 1952, these two actually did some experiments with the bacteria, E. coli, and a bacteriophage. And the experiment proved that DNA and not protein was the factor that influenced it. And the reason we picked bacteria, it's a simple life form, not a whole lot of organelles inside of it. In fact, no organelles inside of it. It does have DNA, not in a nucleus, and it's got a cytoplasm, which does everything a normal cell has to do, just doesn't have the concentration in an organelle or a wrapped organelle inside of it. And then they picked another one, which was a bacteriophage. Bacteriophage actually means bacteria and then to devour, and it's a virus. And they ran 2 different experiments. One, they put irradiated sulfur in the protein code of the virus, thinking that if the protein code actually contained the genetic material, these E. coli bacteria would actually have it, would show up as radioactive, because the radioactive protein coat would go inside the bacterium and that would come out. And the second one, they actually used phosphorus, radioactive phosphorus, and they put it in the DNA strand, trying to see if the DNA actually was the radioactive material. And when they finally injected it into the bacteria, they let them sit there for a while, then they ran them through a centrifuge to separate things. We found out that the protein coat stayed radioactive, and the bacteria, or the bacteria, the E. coli, did not have radioactive material as it went through mitotic divisions. And then the one that actually had the phosphorus got injected into the bacterium, and as it actually got duplicated, that bacterium carried that radioactive material. So they proved that the DNA was the genetic material. Then we have Chargaff. Chargaff actually is given credit for the base pairing. What he did is he actually started looking at DNA, and he found out that adenosine and thymine had the same percentage, and cytosine and guanine had the same percentage. So if we had 15 and 15, which is 30%, we had basically 70%, 35s and 35s, 37 and a halves and 37 and a halves to go to these two. And he actually figured out that the A's and the T's and the C's and the G's were the same. So every time we hit a G, we got a C, and you can see 3 and A's and T's, 2, the hydrogen bonding in between them. That's the section in that spinning object that was in the first slide that didn't touch each other. And we do have the double helix format. And I can go on to the next one. So there are the 2, there are the 3, and you can see that the hydrogen bonds, basically when we split this thing using helicase later on, relatively easy way to split it. We do have 2 strands. We have the normal strand, which is called the 5'3", the first way we actually saw the nucleotide, the monomer for DNA and RNA to format. Then we have a 3'5" side, which is the exact opposite, and that actually has to do with the fact that we have the 5 carbon on this 5 carbon sugar up here, and we have the 3 carbon down there. So if it sits normally, it's a 5'3" strand, and if we flip it upside down, it's a 3'5" strand. Rosalind Franklin Morris Watkins first actually take pictures with X-ray crystallization, and there's the machine, and here's the picture that Rosalind actually took. I got to do something here for a second because I just got a call, and I'm hoping I can end this for a pop. Okay, I'm back. So Rosalind took the picture of the DNA strand, and it actually gave clues to the actual double helix, where you can see the 2 parts of this, the top and the bottom, in focus, and it sort of looks like it actually moves out of focus and into focus, so it looks like it actually spins in a direction. You can see the rungs, the actual nucleotide, the nitrogenous bases, and you can make out, at least for part of it, that these were lined up so you can see the hydrogen bonds inside. They are not given credit for that. They're given credit for actually taking the picture, and I think Mr. Watkins actually took credit for her picture. So, double helix, looking at the picture, here's the actual picture signed by Franklin and Pushing. You can see the paperclip rust stain that's in there. It actually was folded on top, but this is the actual picture that she took. Nobel Prize was given to Watson and Crick and Wilkins and Franklin in 1962. Unfortunately, using X-ray crystallization exposed Rosalind to a lot of X-rays, and she probably got cancer from that job. Then we have the fathers of DNA technology. We have Watson and Crick. They're remembered as the fathers of DNA. Watkins and Franklin have been stuck to historical footnotes, and they came up with the double helix. Here's the actual model that they made way back in the 1950s, and here's the one that I showed at the beginning, which I think would bring them even more awe than the one that they had. Then I threw this one up there as a joke, and it says that when we had somebody describing the DNA molecule, they could say, as you can see from the model, structure is basically, well, a long, twisty, rope laddery type thingy. Okay, so I reviewed DNA here, where we actually have the strand of DNA, and then it actually finds histones and starts winding in on itself, and then it becomes chromatin, and it keeps winding and winding and winding until we actually get a chromosome made of 2 sister chromatids, and all of that stuff is inside the DNA, only visible as a chromosome when we're going through the mitotic division. What does DNA stand for? Deoxyribonucleic acid. Why that's blue, I'm not exactly sure, other than to show that oxygen, deoxygen, should not be there. So it's missing an oxygen. A hydroxyl and ribo sugar has actually changed it to a hydrogen, so it's lost the oxygen, deoxyribo. What is the function of DNA? The function of DNA is it holds the genetic code. All critters are made out of proteins. This is the code for building proteins. And it basically holds the directions for all the proteins a cell needs to make itself. How are the proteins used? For structures, like muscle fibers and enzymes, for transport proteins across the cellular membrane, for hormones, where we actually have one individual talking to another individual of the same species. So pheromones too, where we actually talk about one part of an individual talking to another part of the same individual. What's the DNA molecule made of? Another bad sentence. What parts are there to a DNA molecule? And I wanted the kids to draw it, although I gave it to them on their notes. We have a phosphate group up here. If we had 2 of them, it would be diphosphate. If we had 3, it would be adenosine triphosphate. We have a sugar, in this case it's the deoxyribo, and this is the 5,3 orientation, the normal orientation for a nucleotide. And then we have the nitrogenous base, which is the A's, the T's, the C's, and the G's. What are the 4 nitrogenous bases? Oh, and actually I talked about them being in 2 different groups. And you can see that thymine and adenine are not in the same group, just like cytosine and guanine. And when we get to RNA, we'll actually throw uracil in there because there's no thymine in that language. 4 nitrogenous bases, they're called complementary base pairs. We have adenine and thymine, and I actually have them underlined over here to go along with the greens and the oranges on this side, and guanine and cytosine, and you can see that this is a larger molecule and these are the 2 smaller molecules. DNA nitrogenous base, how do they bear it up? It all has to do with hydrogen bonds, and adenosine and thymine, or adenine and thymine, actually have 2, and cytosine and guanine have 3 hydrogen bonds, so they're bonded a little differently also. How are the DNA molecules shaped? What makes up it's a double helix, which means a twisted ladder. What makes up the sides of the ladder? If you take a look, the base pairs are in the middle, so that means that the sugar and the phosphate make up the backbone, and the nitrogen bases make up the rungs. And I had them look at this picture, so the sugar and the phosphate, one group, and the nitrogenous bases with their hydrogen bonds, the base pairs being the other group. Then we labeled this diagram where we had the phosphate group, the P's. We had the sugar, which make up the backbone. We have the hydrogen bonds holding the 2 nitrogenous bases together, and then we have them all together making, we have the phosphate sugar backbone, we have the nitrogenous base rungs, and throw all the phosphate, the nitrogenous base, and the sugar together, and we get a nucleotide. And I think I am going to call it quits there for 7A Part 1.